Okay, thank you. So yeah, absolutely delighted to be here to tell you a little bit about what we do at the gardens. I want to begin by acknowledging the Gadigal people of the Eora Nation, the traditional custodians of lands that include University of Sydney and the Royal Botanic Gardens, Sydney. I want to pay my respects to elders past and present and acknowledge the continuing connection between Aboriginal people and the lands and waters of New South Wales. So a quick outline for this talk today, but thanks, Travis, you've already mentioned a few of these things. Thanks so much for the introduction. So I'll start by just talking a bit about the Royal Botanic Gardens, what its mission is, with a special focus on the collections that the Gardens holds that in some ways are very pertinent to data. I'll also talk about the research that we do at the Gardens using those data. And then I'll just dive in and talk about 3 projects that exemplify some of the things that we do, although they're drawn specifically from to include things where we have needed to make inferences in ways that weren't necessarily already really easy to access or firmly established. So I guess projects that might exemplify the sorts of things that an organisation like the Gardens and researchers such as those at DARE could maybe collaborate on productively. So I'll introduce those as I go through. So to begin, the Royal Botanic Gardens was established in 1816 for research in botany and horticulture. This makes it the oldest scientific institution in Australia. And when it was established, it occupied the site where it currently does in Sydney, near Woolloomooloo. 2 more gardens were added to the organisation in 1988 and 1989, respectively, at Mount Annan near Campbelltown and at Mount Tomah in the Blue Mountains. And the mission of the gardens, or its objectives of the Royal Botanic Gardens and Domain Trust, is really right there in the legislation that establishes the organisation, and that is to maintain and improve the lands that the gardens are on and the collections of living and preserved plant material that it holds, to increase and disseminate knowledge of Australian plant life, and to advance the study of systematic botany and plant conservation. So already we can see that the collections held by the gardens are at its centre. So I'll talk just a bit more about that, specifically the ones that are kind of interesting for us today from a data perspective, sort of, and that align with the interests of DARE. So one is can you see my cursor if I move it around, just out of curiosity? Okay, thank you. First of all, the living collections. So those are all the plants in the gardens. Most of them are accessioned, so we have information about them. They're in a database. They've been identified by a botanist. So they're an amazing living resource for people to go who are studying a particular group of plants and look at them and the living structures. Our organisation also has a collection of approximately 1.4 million preserved specimens. They look like this picture on the right, which is a specimen of golden wattle, Acacia pycnantha. So this is a massive collection which is housed at or is currently in the process of being moved from the old New South Wales Herbarium at the Sydney site to a new purpose-built facility at Mount Annan, which has great temperature and moisture controls, etc. So those resources are used in research on plants that can be sort of organised into probably 4 main themes. So one is taxonomy, or systematic botany, how plants are related to each other, how we classify them into different groups. Plant pathology, dealing with diseases of plants, which is obviously really important. Horticulture and seed banking, which obviously we need to be heavily engaged with horticulture for growing plants, but also doing research on how to grow plants that are of conservation concern, for instance. And the part of the organisation that I'm most heavily involved with, conservation and restoration, which is housed at the Sydney site in the Research Centre for Ecosystem Resilience. So what I'll mention first, just because it goes so strongly to the themes of the collection, is systematic botany. So basically, if we have a number of different herbarium sheets, such as the ones in the pictures shown here, this just sort of illustrates the value of the collection. People can use the specimens and different structures of the plants to classify them into groups that also hopefully bear a close resemblance to the actual evolutionary history of those groups. So these two plants in the same genus of callistemon hopefully share more similarities to each other in traits that evolved slowly than either does to this Melaleuca, and those three share more than with each other than with this Syzygium or lily-pilly. Another point that I really wanted to make here, though, is that the Gardens has just undertaken the process of digitizing this collection. So all of the specimens were photographed, they were placed on a conveyor belt and moved underneath a high-resolution camera to take very standardized, high-resolution photographs of all of the specimen sheets. So a point that I'd add here for people who are interested in data is that what this sort of represents is a very large collection of specimens, each of which has associated information, including the date of collection, the location at which it was collected, which can be used to access information about the climate of origin. We can sometimes measure traits from specimens, and I'll talk more about that in a moment, but also what the plant was doing at the time of collection to some degree, whether or not it had buds or flowers or fruit. So we call that the phenology of the plant. So that brings me to the first exemplar project that I wanted to talk about, and that is trait measurement from specimens and what we can use that to infer. So just to start conceptually, I just want to talk a little bit about associations between the traits of plants and climate at really broad scales. So globally, leaf size, I read in a citation recently, varies, I think it was 100,000 fold from the biggest to the smallest leaves. So from this Epacris that you can see here on the left, to this, the scale bar of 10 centimeters up in the top right-hand corner here, this enormous leaf. So there's this massive range in variation. And if we knew about associations with climate, that might help do things like inform vegetation models, which are sort of parameterized to know something about how the plants or the vegetation in a particular grid cell responds to changes in environments in terms of the biogeochemical processes that it catalyzes, you might say. So, so in 2017, Wright and others published really sort of a landmark paper on this in relation to leaf size. They did a large literature synthesis that included more than 13,000 combinations of species by sites associating leaf area measurements with environmental variables. What they found was that on the average, larger leaves are associated with greater rainfall, higher temperatures, and lower latitude. So basically larger leaves in the tropics. So this starts to help us understand how plants evolve in different climates, but also, as I mentioned, potentially can be used to do things like parameterized models downstream. I should point out that these are these patterns only sort of, in some cases, become apparent at these global scales. And we can see this immediately if we just wander into the bush and see that any given climate, leaf size varies by some orders of magnitude also. It's just when we look over this massive global domain that these patterns start to manifest. So with collaborators, including Will Cornwall at UNSW and Brendan Wild, an honours student who worked with Will and I last year, we started thinking about how herbaria might augment these global data sets. So, and in particular, it's still quite a lot of effort to measure leaf traits from specimen sheets. So we started to wonder, could we extract traits reliably with machine learning models towards this goal? So I'll just tell you a little bit about what we did on that. So ideally, we would take a sheet and use a trained machine learning model, say a convolutional neural network, to predict leaf instances in terms of their pixels, convert that to centimeters squared. We know the locality of the site of origin. We can extract climate information and look for correlations. So another representation of that, the goal would be to take one of the specimen sheets, label it, draw polygons around the structure that we want to be able to predict, train a convolutional neural network model. I won't go into the details of that very much at all today. Rather, I'll sort of focus on the domain applications, but then the process, of course, would include testing that model on labeled images that were not used in training, which might tell us we need to refine the model a bit further. But at some point, the goal would be to end up with a model that we trust and use it to make predictions for many more images and test associations between that and climate. So we decided to do this within a focal taxon rather than trying to do it across all plants whose leaves can sometimes look wildly different. We decided to start within a genus to try to do it on a more manageable scale. And we chose the genus Syzygium or lily-pilly, which are all around Sydney. You see them often, but a couple of really nice things about this genus for getting started is that they're distributed across a pretty large latitudinal gradient. Along Eastern Australia, Northern Australia, and up into Papua New Guinea. And they also, as you can see from these pictures here, show pretty different leaf sizes across that. So it seemed like a wonderful place to get started for Brendan's honours project. So part of the goal here was to see, because we knew that ultimately we would like to expand this to much broader taxonomic scales, part of the goal was to see how far we could get using relatively modest amounts of training data. That is, we don't want to have to do too much training for a single genus if there are hundreds or thousands of other genera we'd like to apply this to. So Brendan did his training here using just 225 masks drawn on specimens. Just some technical detail. They were the training was done using a model with ResNet 50 architecture implemented in Detectron 2 by Facebook Research. So we then did some testing. You can see the values of precision and recall there, which are not amazing, but are not too bad for the relatively modest amount of effort that had to be invested in training. So this is just a plot showing the areas predicted versus the areas from the hand-labeled data. We can see that it falls really neatly on a one-to-one line for the matched images. One thing that I will note is that we tended to over-predict slightly for the smallest leaves. So we're sort of below the one-to-one line down here, which is one thing that we'd be a little bit concerned about, except that the predictions we were doing were across such a large domain that it's sort of proportionally quite a small amount of error. So just to share the results, this in some ways was a really fun project in that it's had sort of some biological outcomes that sort of emerged during the project, which was really fun, and that sort of highlights some interesting ways that herbaria can contribute to this question in new ways. So Brendan started, so first of all, across the whole data set, across some, I think it was 9,000 leaves, 1,300 specimens, we saw many of the same relationships that had been reported in a global data set by Wright et al. That is, in Syzygium, bigger leaves at low latitudes, relatively high temperatures, and high levels of annual precipitation, which is really interesting and useful, and sort of confirms that that same relationship that was observed globally also occurs within one interesting genus. Brendan went sort of a step further here, though, and started asking questions about what happens within individual species of Syzygium that occur over quite a large range, and for which we had more than 100 specimens. And the results were really inconsistent. Sometimes there was a bit of an association, sometimes there was no association, sometimes even a sort of weak tendency towards a negative association. And so we set out to kind of ask that question a little bit more formally across the whole data set using mixed models. So testing the same associations between leaf size and the climate variables, temperature and rainfall, but putting the identity of the species into that statistical model as a random effect, and found that overall the explanatory power of the model was largely lost. That is, there was, in average terms, while there was a relationship across all of the specimens with these climate variables, on average, there was not an association also within each species, which, one really nice thing about this herbarium data set is it sort of has the resolution to test that hypothesis a little better than has been possible before. So just to summarize some of the outcomes of this first little exemplar study, a combination of herbarium collections and machine learning could potentially massively augment trait databases and contribute to our understanding of associations with climate. Syzygium leaves were bigger in warmer, wetter climates and near the equator, but this variation seems to reside among and not within species, and suggests some further hypotheses kind of around genetics. Does gene flow among populations within a species prevent things from adapting in the same way that disconnected populations can adapt? But of course, there's also a lot of really interesting questions to ask in the future in relation to this about how to do this better. So how do we deploy these methods beyond a single genus, for instance? Really central to this is, what are the limits to training? Of course, if we were to take the model trained for Syzygium and try to predict leaves in Melaleuca or Ficus, would it work? Or is training quite taxonomically specific, or moreover sort of specific just to the visual appearance of the leaves? Of course, also one could do lots of really interesting and fun things for other traits, buds, flowers, fruit, etc. So there's just so much to explore there. It's really exciting. So I want to move on now and talk about something almost completely different. Other than that, it's also something where we've sort of had to come up with different kinds of inferential methods than were immediately available to us. And this is really sort of my core research interest. So this is in conservation genetics and doing optimization to help ex situ conservation efforts. So just some acknowledgements from the beginning here. This research is all done in collaboration with members of the Research Centre for Ecosystem Resilience at the Royal Botanic Gardens, Sydney, also with our colleagues in horticulture at the Australian Botanic Gardens at Mount Annan. This research is also all done in collaboration with partners from the Department of Planning and Environment, often those with responsibility for the conservation of specific threatened or endangered species. We often also pursue this research in collaboration with university partners. I'll acknowledge specific collaborators as I go through, but I just want to underscore that this is all a very big combined effort. So just a few words first on plant conservation. So there are more than 700 plant species in Australia that are at least threatened or endangered. The ones that we see here are just a collection that were believed so insecure in the field, in at least the whole species or populations, that they've been brought in for ex situ conservation treatment. And what this really highlights for me is that there are many species that merit collection and conservation outside of natural settings, and there's few resources for doing this. So whatever we do needs to be really efficient in terms of the number of plants we keep around, or the bench space we use, all of the costs of doing this kind of work. So I want to also just begin, though, by talking about how we manage these ex situ collections from the point of view of genetic risks. So a little bit of theory to begin with on the kinds of risks that endangered populations face from genetic processes and what we do to manage those risks. So populations of endangered species tend to be small, so quite often populations get really small before they're lost entirely. Small populations are at risk of losing a lot of diversity. And one of the possible consequences of this is losing the ability to adapt to future challenges, because genetic diversity is the substrate of evolution and responding evolutionary to fewer challenges. So to illustrate that, I'll ask you to imagine two populations shown by the large circles. Individuals within them are shown by the small circles. The different colors just represent, say, different copies of a gene at a particular place in the genome. So the population shown at the top is more diverse in that it has two different kinds of genes. The population at the bottom is less diverse. Let's imagine sometime in the future a challenge comes along, a change in climate, a new disease. In the population at the top, the frequencies of those different genes can shift in the population. It has evolved adaptively and can persist. The population at the bottom might by chance not have a gene that confers an advantage under the changed condition. It risks population contraction or extinction. So the risk of this to small populations is the loss of adaptive capacity. The goal, the principle that we try to follow in managing that risk is maintaining as much diversity as we can. Second, another risk faced by small populations is inbreeding and inbreeding depression, so loss of fitness associated with inbreeding. And this is because it can, just in small populations, it can be hard to find unrelated individuals for mating. So I'll begin by asking you to imagine a location in the genome where there are two versions of a gene, a wild one, which is just fine, and another one that might be non-functional. There's been a mutation in the gene that causes it to stop working. And this happens in a very particular way. So if we have an individual that has one copy of each, if it has two copies of the good gene, it's fine. If it has a copy of the good gene and a copy of the bad gene, it's also fine. One good copy can do the work of two. But if it gets two copies of the bad gene, suddenly it's very unfit. The individual might not be viable. So we call this a deleterious recessive. Deleterious recessives tend to be very rare in populations. So the probability of two individuals that carry one coming together to mate to produce individuals that have two copies of the bad gene is very low. So these negative consequences of this bad copy of the gene are rarely realized. However, as soon as we start to get a little bit of inbreeding, as we see in this pedigree here, say for instance, if two individuals mate, one of them has the bad gene, and then two offspring from that pairing mate, suddenly it starts to become quite probable that we produce individuals that have two copies of the bad gene and that suffer these negative consequences, this unfitness of inbreeding. So a risk to small populations, including in an ex situ setting, is inbreeding depression. Our goal is to avoid inbreeding, to try to reduce the amount of genetic relatedness in a collection of individuals. So let's now see how we apply that to a particular species that in this case, New South Wales Department of Planning and Environment officers were trying to conserve. So this is a plant taxon who, at the time at least, it was all of the individuals of it that we knew about occurred in one very small area in Western Sydney. So it was at very high risk of extinction from a single catastrophic event. A fire could take out the whole plant taxon very, very quickly. So the threatened species officer responsible, Ahmad, decided to undertake a translocation, so to take individuals from the wild population and plant them in another location. And so the procedure for doing that looked a little bit like this. The idea was to sample the individuals from the natural population. I'll add here that at this point we took samples for DNA, so we genotyped them. The idea then was to grow all of the plants collected from the field at Mount Annan, grown by our colleagues there in the horticulture teams at Mount Annan, to grow up a number of different genetic copies of different individuals and to plant those out at a translocation site matched for climate and soil. So to try to bring them at first into an ex situ setting in the nurseries at the gardens, but on their way to hopefully being planted out into a self-sustaining population in another natural setting. So just to talk about how we used some genetics to inform that, and then how we use optimization methods to try to inform that. So we got our genetic data, and these can be thought of as looking at like this. So we have a number of individuals here. We got genetic data for them at these, say, 4 different loci, these 4 different locations in the genome. I'll remind you that a genome is just a big long string of A, C's, G's, and T's. Each individual has 2 copies of its genome, though, one that it inherits from each of its parents. So at a given location in the genome, this individual might have an AA, this individual might have an AA also, this individual has an AG, so it is heterozygous. This individual and this individual each have a GG. There are different bases or different letters at different locations, but the same things apply. The first thing that leaps out is that these 2 individuals on the left are identical at all of the loci, and we might have data sets of 4 or 10,000 of these loci, so quite a lot of data to make inferences about how things are related. But the first thing we can see is that these 2 individuals over here are identical. If that's true over a very large number of loci, we might conclude that they're genetically identical. They might have reproduced vegetatively in the field. So immediately we know that we might only need to keep 1 copy of that in the nursery, so that we don't, so that we can sort of rationalize, and it goes to cost in a way, just paring back the collection to all of the unique individuals. Then when we do the translocation, we can start doing translocations in ways that we can have translocation populations whose genetic composition reflects those goals that I mentioned earlier of maximizing genetic diversity and minimizing the mean relatedness of the individuals. So this is what the data look like. These are the, I think, roughly 48 genetically individuals that were sampled from the site. In each cell of this heat map shows the level of kinship between the individuals. So a kinship value of 0.5 shown by red means genetically identical. This lighter, very light or cream color is no relationship whatsoever. This little cluster of individuals up here were sampled deliberately. They were connected to each other, so we thought they were clones. They in fact were, but that sort of was our validation that we were able to detect clonality where it existed. So now the question is, if we are going to take these roughly 48 individuals and amplify it by taking cuttings, different numbers of cuttings of these genetically unique individuals, for a total translocation of population of 300, which is the number that can be accommodated at the site, what number of each individual do we take? And a simulated annealing algorithm is probably a really good fit for this problem, but it also, we had some other constraints in that we weren't completely unconstrained in the number of individuals of the number of copies of each genetic individual that we had. So we had to re-implement a simulated annealing algorithm in ways that suited our use case. But basically, it took our kinship matrix that I just showed you, the number of individuals that we wanted to plant, and our objective function, which was to minimize the mean relatedness among the individuals, and told us how many genetic copies of each individuals we should plant at a translocation site to meet our objective. So that's what we did in collaboration with threatened species officers and others for Herbatia puberula subspecies glabrescens. We've implemented similar things for other species, such as Eucalyptus species Cattai, which was undertaken again with a threatened species officer, Anwar Lee, and others from my organisation. And there the objectives were a little bit different for Eucalyptus species Cattai. There we were working with a eucalypt, so a tree that occurs at a number of sites in Western Sydney, but one of those sites was unavoidably going to be lost due to development. And so the idea there was, how many seedlings do we need to collect from the trees at the site that's going to be lost, and how do we choose which seedlings to plant at a translocation site in order to capture as much of the diversity as possible that existed at that site, at the site that was essentially condemned. So we implemented a method that was much the same as the one that I showed you for Herbatia, except in this case, we didn't minimize kinship. These on average were a little bit less related to each other. We just maximized a measure of genetic diversity. But then we asked for collections that we optimized, what proportion of all of the genetic variation that we observed in the mother trees were we able to get in different collections of seedlings? And that's shown in the plot that you see here. We have different possible candidate collections shown along the x-axis here, with different numbers of individuals. And on the y-axis, we see the proportions of alleles that we were able to capture. And we did this as an exercise just to see how well our optimization methods were working. So in the black dots, we see populations that we'd chosen on the basis of an optimization similar to the one that I just showed you. And in the blue dots, we see the mean of, I think it was 100 random draws from the same set of starting individuals. And what we see is that if we take a large enough sampling of seedlings, we basically are capturing on the order of 95% of the variation that was observed in the trees at the site. But if we took a substantially smaller set of individuals for a population to try to preserve that diversity, we had to be much more careful about which individuals we chose. The random performed much more poorly than the optimized populations in that case. So I just want to sort of move on now to something where we're possibly talking about slightly more advanced optimization and inferential methods. And so it might start to accord more with the interests of people from DARE than really sort of simple, I guess, simulated annealing from, so now we're starting to think, well, what if we have more than one objective? What if we want to try to maximize genetic diversity, but we're also facing a situation where we can't just maximize genetic diversity because we also have to impose selection. We also, for instance, some of the individuals have a trait that we really want to promote in the collection, such as disease resistance, or another trait that we really want to, whose representation we want to reduce in the population. So we started sort of working on idealized examples of that with the Herbatia data, where we just randomly introduce deleterious traits or deleterious alleles to the collection and try to characterize the trade-off between minimizing pairwise kinship, which is as the values move to the right on the x-axis, and an index of purging, so the degree to which we have gotten rid of a bad allele, like a deleterious recessive, on the y-axis, to draw the trade-off surface between those two sometimes conflicting objectives. So I guess one thing that I would say here is that the algorithm that I used to do that was probably really terrible and inefficient, and is probably an avenue for great improvement. But from an applied perspective, it's really interesting. I guess what we have here is a family of possible solutions, each of which represents a different candidate translocation population or ex situ collection, and then it's up to the manager, the person who's making the decisions about conserving this species, to sort of weigh those potentially conflicting objectives according to their judgments about what is more important, trying to select against, to select for disease resistance, say, or trying to manage the diversity. And we have a number of places where we're currently looking at applying these kinds of methods. One that I want to draw people's attention to is species that are critically endangered to the impacts of the fungal pathogen myrtle rust. So that's a fungal disease of plants that arrived in Australia probably only around 12 years ago. It affects plants in the family Myrtaceae to different extents, some not at all. Some show a mixture of resistance and susceptibility. Others, such as the moist forest species that you can see here, Rhodamnia rubescens and Rhodomyrtus sidioides, are impacted really heavily and are both critically endangered and on a rapid trajectory towards extinction, again with partners from the Department of Planning and Industry, other partners from universities. We've been at the gardens, our colleagues at Mount Annan have been maintaining a collection of these plants, but we're starting to face some of these choices. How do we try to manage diversity and any disease resistance that we happen to observe at the same time, to try not to lose all of the adaptive capacity within the species, but also manage any resistance that we do observe? So some really pressing challenges there, and sort of optimization problems, but with applications that are ready to be pressed into service. So the last thing I want to talk about, and I'll just talk about quite quickly, because I realize I'm probably going to rapidly run out of time, is one that I just found really fun and thought might be sort of interest to folks from DARE. Here we're trying to infer information about the mating systems of plants from genomic data, but we're going to do it using methods that were similar to those that we used for the machine vision study on plants. So the idea is, in cases where we felt that we were starting to sort of come up against some of the limitations of the existing methods for making these kinds of inferences from genomic data, we essentially turned them into images and tried to use machine vision methods to make similar kinds of inferences. So the question here that might be asked in a more general way is a particular plant producing its offspring by outcrossing or by selfing? This question influences some conservation and restoration actions that we might undertake. For instance, it tells us whether or not the plant is likely to be inbreeding and will lead to inbreeding depression, also how much it will mate with closely related individuals in an ex situ setting. But the data here are from a study that was led by Patricia Lou Irving and Marlene van der Merwe from the Gardens. with a number of other collaborators from the gardens. The data are for 2 Hakea species, which are plants in the Proteaceae. There's a picture of one here. It has beautiful flowers. They are very, very spiky. If you've been bushwalking around Sydney and have been spiked by a plant, it was possibly one of these. They're incredibly spiky. 2 species that are very similar to look at, have similar ranges. We were interested to ask whether they're also similar in terms of their popgen. So the idea here was to collect seeds from 10 plants at each of 6 populations, also in the context of a broader population genetic study, so of more plants but without the seeds, from a larger collection of sites across the range. But basically we did genotyping, just like I mentioned earlier. And the idea was to infer rates of selfing from genetic data. So just to talk a little bit more about what selfing is, let's imagine a plant, Hakea sericea in this case, that you can see on the left. We take a seed from it. As I mentioned, at each variable location in the genome, or at each location in the genome, we have 2 alleles in a diploid plant. It has inherited one of these from the mother, so in this case, one of these from the plant where we got the seed, and the other from another parent that provided the pollen. However, the way that we often think about this in this context, where we've gone and gotten a seed from a plant, one allele has come from the parent plant that we know about because we took the seed from it. The other probably has inherited alleles at their sort of representative proportions at which they occur at that locus in the pollen that was sort of raining in on the plant, either distributed by pollinators or by the wind. So there's some characteristic allele frequency in the population. So that's what happens if the plant is outcrossing, if it's mating with unrelated individuals and acquiring alleles basically at their frequencies in the population. However, some plants do selfing, and that is where the same plant that we collected the seed from also produced the pollen that fertilized that seed. So this has a number of consequences. One, of course, it's a pretty extreme form of inbreeding. But in this case, the 2 alleles that it gets is the 2 alleles in the seedling is each represents a random draw from the 2 alleles in the same parent plant. So if we think about this from the perspective of now we're going to do a seed collection and use the seeds to plant out for ecological restoration, we face a lot of questions about how we should do that. So when we go to a particular site, how many seeds should we get from the same population, and how many should we take from the same mother plants? Just because in some cases they're very tall trees, it can be one might want to, for the sake of costs, to minimize the number of different plants from which one acquires the seeds. But as you can see, if that plant has been selfing, we get the alleles in the collection of seeds from a much smaller number of donors, pollen donors, whereas if it's been outcrossing, the alleles that we get more reflect the whole population than just that single individual. So this is where this question sort of affects some of the restoration decisions that we make. However, so far we've just talked about selfing and outcrossing with unrelated individuals, but there's a spectrum of other levels of inbreeding in between. And so there I'll ask you to imagine that the plant from which we've collected the seed a couple of years ago produced an offspring by outcrossing. That seed fell nearby, an individual grew up, and now it's the seed that we've collected might have crossed with that plant. So now instead of getting 2 alleles, one from the parent plant and one from the population's representative allele frequencies, there is some probability that the second allele was also essentially by sort of identity by descent, also drawn from the mother plant in a way that reflects the level of relatedness between the mother plant and the plant that provided the pollen. So basically, we end up with a data set where we have the genotype for a mother plant, the genotype for a seed. We can also measure the population allele frequencies. What we want to know is whether or not that seed was produced by selfing, outcrossing, or some level of biparental inbreeding in between. So we can think of it as just the level of relatedness between the mother plant and the plant that produced the pollen on a scale from 0, no relatedness, total outcrossing, to 1, total selfing. So basically, again, just to reiterate, what we have is data consisting of population allele frequencies, a genotype for a mother plant, and a genotype for a seed. And we want to infer the selfing rate that produced those genotypes. We have this, however, for, in this particular case, some thousands of different loci in the genome. And we want to infer the selfing rate. There are likelihood approaches to do this that were developed by Kermit Ritland in the 90s. However, those are implemented in software packages that only accommodate sort of usually hundreds of loci, which is the number, like there's, I should mention in the backdrop to all of this is our capacity to generate these genomic data sets has exploded from dozens of genetic markers to thousands or tens of thousands in a decade or so. And in some ways, the statistical inferential packages haven't quite caught up to that yet. So basically rather than using the state-of-the-art likelihood methods, we re-implemented a simple method of moments estimator. So I wanted to see how that worked for our data set. And we were able to test that with simulations. So we could say, we know that we have these population allele frequencies. This is our maternal genotype at different levels of selfing drawn from a frequency distribution. We can simulate the production of different seedlings. One other thing I should mention here, though, is that in addition, we know that our genotypes are measured with some amount of error. So we can also build those error rates into simulations. In particular, we think from technical replicates that errors are at low rates, probably on the order of 0.5% or 1/3%, but we think we're most likely to encounter errors where there's really a heterozygote, say an AT, that should be an AA or a TT. We miss one of the alleles that is really there in a heterozygous individual. So really quickly, I just want to mention that we were able to confirm that the simple method of moments estimator on a data set of this size with prescribed levels of selfing in the simulations, if we applied that method of moments estimator, it actually did quite well. If we simulated with selfing rate of 0, we tended to estimate selfing rates of 0 with the estimator. However, we also know that in the pipeline are genomic methods that will make these likelihood methods probably quite intractable. For instance, where we know the order of the genotypes along chromosomes. So we also thought, let's try to extend this using CNNs. So, sorry, I realize I'm running out of time. I'll just try to step it up a little bit. So here our training data are essentially images where we take those genotypes and turn them into numbers. Population allele frequencies, maternal genotypes, which are 0, 1, or 2 copies of a reference allele. The same for the seeds. These simulations have been produced at prescribed levels of selfing, which become the labels. We can train a CNN model, a convolutional neural network on essentially these images, if we want to think of them that way. Then we can present that model with the real data and get predictions. So the first thing I should mention is that for different individuals in our real data set, the estimates of selfing for the method of moments estimator on the x-axis and the CNN were really quite closely concordant. So it seemed to be working quite well, but But as I said, hopefully we can also extend it in various ways. I won't go into it, but it also has some nice ways of sort of testing the limits of the inference. So getting error rates estimates for each individual based on just, sorry, I won't go into it, I'm running out of time. But the other thing that I just wanted to mention really quickly is that these two different species, Hakea sericea and Hakea teretifolia, the different seedlings from them, or the different mother plants rather, showed quite different selfing rates. So in Hakea sericea, there was pretty high levels of selfing across the board, in blue here. In red is Hakea teretifolia, estimated by method of moments and the CNN. And there's quite a sort of bimodality in Hakea teretifolia. So some individuals selfed, some outcrossed. And this was totally concordant with levels of just population genetic diversity observed across the whole data set. Hakea sericea, which was selfing more, showed less diversity in terms of expected heterozygosity, et cetera. So yeah, it seemed that the CNN method seems to show some promise. So basically, to conclude, three really brief things. Sorry I ran out of time so badly, but at the Royal Botanic Gardens, we've got lots of data, some really fun inferential problems that have direct applications and an enthusiasm for collaboration. Please get in touch if you're interested. It probably goes without saying that machine learning has massive potential for research in extending inferences in plant biodiversity. The third thing that I wanted to say is in some way tangential, but I just also wanted to highlight that myrtle rust, which I just touched on briefly, is a really important conservation threat for Australian plants and probably deserves wider recognition at this time. So that's three things to take away. Again, I'll just put up a list of acknowledgements. Thank you so much for your time and attention, and sorry to run a bit over time.